Okay, we reached the last part of our lecture series on allelic interactions, things we're calling more allelic interactions. This is part 3 of the second set, the allelic interactions 2. And what we're going to do here is work a couple of problems, okay? So we're going to start out with a fairly simple problem in the sense that what we've done here is we have basically extracted all the words out, right? So this might be some long paragraph that you have to read, but at the end of the paragraph, this is what you get, okay? You get some progeny. These would be F1 progeny, okay? And they're sitting here, so there's some parents that produce these. You don't know anything about them. You don't really know anything about any of this. So you start thinking, well, what am I supposed to do? Well, I'm supposed to diagram, right? That's what the way you work genetics problems. And in doing that, here are the questions. The questions are, what is the mode of inheritance for each of the traits that might be involved? Uh, then what are the genotypes of the parents that produce these, as well as their phenotypes? And then what are the genotypes of these progeny? What is the genotype of a medium red in this case, okay? And then to do this, you need to develop the segregation ratios and, of course, the assortment ratios, okay? Now remember, no matter how complex the mode of inheritance gets, all genetic crosses are A times B gives us C. And we're working either with A and B, as we've done in some recent problems, or we're working with C, or we're working some combination. In this case, we're working with C down here, right? This is C. We've done the experiment, we've got the answer, and we're trying to backtrack to figure out what A and B were, right? Now, it's not hard. Just let Mendel do all the work, okay? Mendel says you can, if you've got 2 traits, 3 traits, 5 traits, whatever, you can treat each one of those independently and then put them back together to figure out the final answer. So if we think about it, if we go to trait 1, which is height, we see that we've got some tall ones, some mediums, and some short ones. So we would just write that down, okay? We would then say, okay, how many, trait 2, what is it? Has it got 3 things? No, it just seems to have red and blue and red and blue and red and blue. So we'd write that down, okay? Then we would go count them up. We would, because of Mendel, we count up the talls. We don't care whether they're red or blue or not. We just count the talls. So 41 plus 19. Count up the mediums. Don't care whether they're blue or red. We just count them up, right? Same thing for shorts. I did this. You should always check my math because sometimes I don't do it right, but I get 60 talls, about 119 mediums, and about 59 shorts. Then we go count up the reds. We don't care whether they're tall, medium, or short. We just count all the reds. So 41, 79, 39, and then all the blues, okay? When I do that, I look like I get 159 to 79, right? Okay, so what we're gonna do now, we wanna figure out all this information. What's the mode of inheritance? What are the genotypes? What produced all this? Well, we've got the information we need to do that. First off, for the first trait, height, we see that the best fitting ratio we could come up with is about a 1 to 2 to 1, and that's pretty perfect, right? It would be 60 to 120 to 60 would be a perfect fit. So this is as close as we're going to get right here, okay? So we would say that the talls are 1, the mediums come with a 2, and the shorts are 1. So there's 2 mediums for every 1 short, 2 mediums for every 1 tall. All right, so think about it. The genotypes that are associated here, medium gets a hit, right? Always the 2 is with heterozygotes, and then homozygotes are spread out to the other side. We don't care where we put them. We just make sure this is a homozygote, that's a homozygote for the alternate allele, and that this is a heterozygote. So we'll just do something simple, something we've done before. We'll make the tall big A, can make it TT, doesn't matter. Short is little a, can make it little s, and then these would be TS, right? Something like that. Whatever you want to do, doesn't matter. What produced this, right? Well, what mode of inheritance is involved? It's incomplete dominance, right? I mean, I think a pretty perfect example. You've got something that's true breeding that's really tall and true breeding that's really short, and something comes out in the middle, the more common thing, that's incomplete dominance, right? No evidence of codominance because you'd have a tall and a short at the same time. So incomplete dominance would probably be my best answer. A 1 to 2 to 1 ratio, doesn't matter mode of inheritance. Go back to Mendel, right? Keep picking this sheet up. Look here, what will give me a 1 to 2 to 1 ratio? Only one cross, right? A het times het, monohybrid cross. So what I'm saying is you get this. This is the cross that produced it. All right, now let's forget height and go worry about color. All right, we had 159 to 79. That's about, if you start doing division, I mean, think about what you could have. You could have a 1 to 1 ratio, clearly not that. 3-to-1 ratio, let's get look untouchy. It looks like it's about a 2-to-1. In fact, you could divide the small number into the large one and just see what it is. It's almost perfectly 2. So this is a 2-to-1 ratio. Not surprising since this lecture series has been about recessive lethals, but that's what it is, okay? 2-to-1, best fit that we're going to find for that. And these are fairly close numbers to what you'll probably see on tests. You know, we try to make it where you don't have to sit there and guess too much to figure it out. All right. Now, genotypically, the 2 always goes with the hets, right? So these are going to be heterozygous reds and homozygous for something blues. We don't care what you call them. I'm going to make it kind of fit stuff we've earlier done. So we'll have this be the heterozygous for the big B, little b, and this be a homozygote. Blue will be for the blue allele, little b, okay? I've noticed that I've put this big B as red because what we're dealing with here, because of this signature ratio, is a recessive lethal, all right? This is a classic recessive lethal. All right, we got a 2-to-1 ratio. The only way we can get a 2-to-1 ratio is to genetically produce a 1 to 2-to-1 and just lose one of the ones, one or the other, okay? Because we don't know which, we just lose them. The only thing that will produce a 1 to 2-to-1 is a heterozygote times heterozygote cross, right? All right. So what else does it tell us? It tells the mode of inheritance. It tells the genotypes. It tells me what the parents were from each of these, right? So if I think about what the parents are, well, one parent is big A little a, and the other one's big A little a. So that means they're mediums. So I must have 2 mediums here, right? Okay. And the other side, they are big B, little b, which means that they're going to be red. So this is a classic dihybrid, you know, double heterozygous cross. Both parents are double heterozygous. Both parents are medium red in this particular case, okay? Now remember, these are the segregation ratios, right? Segregation ratios is what you get directly off of Mendel's Big Six chart. That's for one trait. Okay, so each one of these is a segregation ratio. We want to produce the final phenotypic ratio up here. That is the assortment ratio. That's when you multiply 2 or more of these segregation ratios together. So in our case, every time we have a tall, we put down a 1. Every time we have a medium, we put down a 2. And every time we have a short, we put down a 1. If you want to put them on top of these so that you can remember it, that's fine. Okay, multiply them out. We end up with a 2 to 1 to 4 to 2 to 2 to 1. This is our final assortment ratio that is phenotypic, right? The way it looks, okay? This is our phenotypic ratio. Now, what are we missing? Well, what we're missing here are, notice this, normally when we cross a dihybrid het times a dihybrid het, we get 16 parts, total parts. As I told you, burn that 16 in your brain. Well, here, 2 plus 1 plus 4 plus 2 to 2 to 1 is only 12. Where are the other ones? Well, they're dead, right? So it would be plus 4, which would be some that are not seen. You don't know what color they are, you don't know what height they are or whatever, but they're not seen, all right? Okay, so this is our final phenotypic ratio. And if you were asked to do a problem, as we're going to do in a moment, you'd say, okay, I expect 2/12 to be tall reds, 1/12 to be tall blues, 4/12, you know, to be medium red, or whatever. Okay, that's a final assortment ratio. Didn't have to memorize it, didn't have to try that. You always generate it from the segregation ratios. All right, let's try another problem. Here's what you might see on a test. Oh my goodness, I've got to read, and they're asking all this stuff, and I've got to figure it out. And I only want to spend 3 or 4 minutes on this problem. You know, no, don't worry about it. Okay, all of this stuff at the top is just telling you some information that you can extract given information from. Okay, and what is it giving you? It's telling you up here that you've got this N allele, okay, and you've got this NN allele, and you got this MN. So N allele causes things to be hairy. MN says they are haloed, and then little n is that they're normal, okay, normal wool. So you've got that. I just wrote it down. So that first 3 sentences all contract down to this. The next one says, okay, I've got some kind of lethal problem going on here, where homozygous G's give me lethality, okay? I've got heterozygotes produce gray fleece, so the gray color, and the little g's produce black. So I've told you what alleles to use. It's not that hard, okay? Now, what do you think? I bet you're going to make you do a cross, right? Some kind of cross. So you'd be thinking of A times B, going to be C, Okay, so guaranteed that's what's going to happen. Sure enough, if two halo gray individuals are mated, what do I get? Two halo gray individuals. Well, what is that? It's easy. Here's a halo, there's a gray. Guess what? I'm doing a classic dihybrid F1 cross, right? I'm sitting here and I'm saying what I'm crossing is a halo gray, halo gray, halo gray, and I want to know what I'm going to get. I need to diagram it. So this is the same as big A, big B, little b. Nothing different except different letters, right? Crossing with this. All right, I let Mendel do all the work, and it says as far as the halo or the sort of hair color shape locus, I'm going to get a 1 to 2 to 1, right? Okay, because obviously that is some kind of incompletely dominant thing going on here, or codominant, whichever you want to call it, right there. Okay, and for the other allele, I'm going to get a 1 to 2 to 1 as well, right? So for this locus, rather, for the gray, I expect some to be gray, some to be black, but look here. I've got some combo here that give me recessive lethals. So these are going to be lethals, right? All right, I just put all those together. I just put the big N with big G, and big N with little g, and kind of write them all down. And when I do, I get all this stuff, okay? Right? I get 1 times 2 is 2. 2 times 2 is 4. 1 times 2, so I've just written them all down. Everybody that's got big N is hairy. So I've got a hairy, you know. Everybody that's got this one right here is halo and normal, and these are grays and blacks. That's what I get right there, okay? Now, when you make this cross, remember dihybrid het cross times dihybrid het, you expect 16 parts. We, of course, don't see 16 parts because some of them have died off. So there are 4 lethals, 4 dead ones that belong in here, and they could be of various combinations. You could figure out what they should have been by back crossing these and looking, but that's what they should be, okay? All right. So then there's a series of questions. Once you get this, you saw, remember that original thing, there was a whole bunch of questions, right? So here's one of the questions. What proportion of the live progeny, got to read these very carefully, carry the lethal allele? In other words, how many of them have a big G, right? Of the live progeny. So you're only worried about this pile right here, okay? You don't have to worry about lethals at this moment, right? So that's got a big G, that's got a big G, that's got a big G. I would add 2 plus 2, 4 plus 2 up, okay? And I would see that I've got 8 parts out of a total of 12, not 16, but 12, because it only asks about the live. So 8/12 are 2/3. So about 2/3 of my progeny are going to be carriers for the lethal allele, right? They're going to have the capability of passing it to their progeny. The next question I think was, what proportion of the live progeny with halo hairs would carry the lethal allele? Okay, so we're back over here dealing with this lethal. So we're saying, what are we talking about? We're talking about I need to be haloed, okay? So halo has to be the heterozygous condition for the N locus, and I have to be carrying this lethal, which means I got to have the big G. So basically I got to look like that. Of the options I have here, like that, okay? Now, be careful when you read this, okay? Of all the live progeny, there's 4 of them that have this genotype. So that would be 4/12. Of the live progenies who also have halo hairs, you're only dealing with these guys and these guys down here, okay? So you're saying, I just want to deal with halo hairs and I want them to be alive. Well, these guys have halo, these have halo, right? Doesn't matter what the color. So in that case, it's actually 4/6 or 2/3. So you just have to be a little careful when you read the problem and make sure that you pick the right parts of it, right? And that same thing applies to the last question. What proportion of all the progeny we expect to have genotype so-and-so, okay? What proportion? All the offspring. That means we're now including the lethals. We're just simply saying, if you make this cross up here, and you do both of these crosses, how many of them are going to have this genotype? Okay, so now you're out of 16 parts, right? Well, you look up here and you say, okay, I need this genotype right here, halo black, okay? That's what they're going to look like. That's what the part of the question is. And I'm going to have 2/16 this time. So this time we're having to be careful. So the problems are, I mean, they're trivial to do in the sense of setting them up. They're just Mendelian problems. You just lose. I mean, it could have been the big G's or the little G's. I mean, we set it up so it forced you to be the big G's, but you could have picked the little G's if you're doing it yourself. You expect 16 parts, you only get 12 living, and you end up this, okay? Play with this. Take some recessive lethals and throw them in there with dominant. Just try different things with this. Maybe do a trihybrid where one of them is a recessive lethal, one of them is a dominant, and one of them is a codominant, and see what you get. Just play with it. That's how we make up problems on tests. You know, it's the way we work with it is simply like that. And this is a pretty good example of a test question. Because you have to go through all these calculations, and then we're going to ask you, we're going to pull some things out of it and ask you about those, right? We may not ask you to show the work of the whole things because it takes a while to type that on the web, but we're likely to come back and say, oh, tell me this part of it, okay? Which means you actually have to do it and work through it. All right. Well, that concludes this sort of part where we're talking about some other modes of inheritance. We're going to take a slight side trip with the next couple of lectures and use all these modes of inheritance, but introduce a new idea and or something that's important to us, and that's called multiple alleles. And then once we get through with that, we'll pull back and start introducing yet more modes of inheritance.